Good morning, students. Today we are going to start with a new chapter, Seeds, Structure and Germination. In this chapter, we will be talking about structure of dicot and monocot seeds, germination of seeds, types of germination, conditions necessary for germination and experiments. What is a seed? A seed is a ripened ovule. It contains an embryo which develop it into a new plant. Formation of seed, embryo, endosperm formation are all studied in the previous chapter. Revise that part thoroughly. Seeds vary in their size and shape. Some of them they are very small, some are large. Some examples are some seeds they are very small. Poppy seed is an example. Orchid seeds are also examples for small seeds. Seeds weighing less than 25 gram were grouped as small seed, while seeds weighing above 25 grams were classified as large seeds. Now you can classify which one is a small seed and which one is a large seed. Largest seed. So seeds vary in their size and shape. Next, classification of seeds. Seeds are classified on the basis of number of cotyledons. The seeds are classified into monocotyledon and dicotyledon. Monocotyledon, mono means single. Seeds with only one cotyledon, they are called monocotyledon. Examples, maize and grass. Dicotyledons, di means two and two cotyledons are present. Bean seed is an example. Another classification on the basis of endosperm. In some seeds, endosperm present and cotyledons are thin and membranous. They are called albuminous or endospermic seeds. But in some seeds, cotyledons are thick and fleshy. Endosperm absent. They are called exalbuminous or non-endospermic seeds. Albuminous seeds and exalbuminous seeds are many examples in monocot category and dicot category. Some of them they are given in this table. Observe these examples and learn these examples. More examples are given in your book. Now we will study the structure of a bean seed. Kidney beans are dicotyledonous seeds and they are non-endospermic. Dicotyledon means two cotyledons are present. And the seed covered with a seed coat. The pink colored or red colored coat, outer covering, that is the seed coat. And the seed coat consists of two membranous covering, an outer testa and an inner tegmen. The seed coat protects the embryo from adverse climatic conditions and helps to prevent from drying out. On the concave side, a whitish scar present called hilum. This is the spot where the ovule attached to the ovary. Ovule attached to the ovary wall through placenta, that is the hilum. Near to that hilum, a minute opening present, that is called a micropyle. What are the functions of micropyle? It allows diffusion of respiratory gases during seed germination. And also, seeds absorb water mainly through this micropyle. Inside the seed, the embryo in an inactive condition. The inactive embryo become active in presence of water. So these are the functions of micropyle. Micropyle allow exchange of respiratory gases. Oxygen entered inside and carbon dioxide is coming out. Another function, water enters inside the seed during germination. Now, next, the internal structure of a bean seed. When we remove the seed coat, below the seed coat, two cotyledons are present. The cotyledons are the reserved food material for the growing embryo and also protect the embryo. In between the two cotyledons, a baby plant or an embryo is present. The embryo consists of two parts, a radicle and a plumule. And the embryo is connected with the cotyledon with the help of embryonic axis. The plumule with two short leaves. Later on, it develops it into shoot system. The lower part, that is a radicle, and the radicle changes it into root. So this is the structure of a dicotyledonous seed. This kidney beans, the hilum is very clearly visible. The whitish scar present there in the concave side of the bean seed. And this is the picture that which shows the seed attached to the ovary wall through placenta. That is a function of hilum. Hilum which helps to connect seeds with ovary wall. The outer membranous covering, that is testa, and inner to the testa a tegmen present. Now we will learn the structure of a maize grain. Maize grain, it is a monocotyledonous and endospermic grain. Monocotyledon means only one cotyledon present. Why it is called a grain? We use the word maize grain, rice grain, wheat grain. Why? It is a fruit in which the pericarp or fruit wall fused with the seed coat to form a protective layer. This diagram, that which shows the external view of maize grain along with the longitudinal section of maize grain. And here in the external view of maize grain, one side of the grain with a light colored oval area, that is the location of the embryo, and the remaining large part, that is the endosperm, the reserved food material for the growing embryo. The endosperm and the embryonic part separated with the help of a thin epithelial layer. Epithelial layer is very clearly visible in this diagram. The epithelial layer, that which separate endosperm with embryonic part, and it secretes some enzymes and dissolve the endosperm for the embryo. The endosperm is lined with a proteinaceous layer, and that layer is called alluron layer. The embryo consists of a single cotyledon, that is why it is monocotyledon, and that part is called scutellum. A radicle and a plumule present. Radicle lies towards the pointed side of the grain, and the radicle is covered with a membranous covering, that is called coleorhiza. What is coleorhiza? Coleorhiza is the membranous covering of radicle. Plumule towards the upper side and covered with a membranous covering, that is called coleoptile. What is coleoptile? Coleoptile is the membranous covering of the plumule in maize seed. So coleorhiza and coleoptile, these are the two membranous covering of radicle and plumule present in case of a maize grain. This is the structure of a maize grain. Next, now compare the difference between a bean seed and a maize grain. mainly a dicotyledonous seed and a monocotyledonous grain. Bean seed, two cotyledons are present, endosperm absent, embryo is large, plumule large and leaves are folded, hilum and micropyle clearly visible, seed separately contained in the fruit called pod. Check all these characteristics. Now, for maize grain, only one cotyledon is present, a large endosperm present, small embryo, plumule small and leaves are rolled, hilum and micropyle not visible, seed wall and fruit wall fused to form a single grain with no separate seeds. So that is the difference between a bean seed and a maize grain, or the difference between a monocotyledonous seed and a dicotyledonous seed. I hope you understood the structure of dicot and monocotyledonous seed. Read book thoroughly, practice all these diagrams, and next class with another topic. Take care. Thank you.